I'm Tom Kornberg from UCSF. In these presentations, I'll be talking about how cells signal to each other, specifically about the mechanism of paracrine signaling. That is, the cell-cell communication that results when signals produced by a cell induce changes in nearby cells. The title of these presentations I'll give as this: Signaling at a Distance, Communicating by Touch. In the first presentation, there will be two. The first presentation will provide a conceptual background to paracrine signaling, so it's a very general background. The second will describe recent research that gives us experimental evidence for a mechanism of paracrine signaling that has very general and interesting implications for many different biological contexts. I'd like to begin by referring to a paper from a graduate student at Columbia University who published this paper in 1909. And as you can see, the title of this paper is The Production of New Hydranths in Hydra by the Insertion of Small Grafts. And I show this slide in part to also illustrate how fashion changes in science. And I'd like to read this first paragraph for you. So this graduate student, her name is Ethel Nicholson Brown. And she was a graduate student of E.B. Wilson. Now, the name E.B. Wilson may be familiar to you. It turns out he wrote one of the most famous texts in all of biology, textbooks called The Cell. As you can see, he lived between 1856 and 1937, and he's credited with many major discoveries in cell biology. He's considered to be the premier and first most important American cell biologist. And in fact, to this day, the American Society of Cell Biology grants an annual award, a medal in his name, the E.B. Wilson Medal. But let's read from this first paragraph. I think you'll enjoy this. Here's what she says in her paper. During the winters of 1906 to 1908, I carried out some experiments in grafting Hydra viridis for the purpose of throwing more light on the factors concerned in regeneration. The work was done at the suggestion of Professor Morgan, also a professor at Columbia University, whom I sincerely thank for his kind interest and help. So who is Professor Morgan? That's T.H. Morgan. So he ran the famous fly room at Columbia University, which is responsible for so much of the foundation of genetics that we know today. They discovered and showed that genes are in chromosomes, which are the units of heredity, one of the many major discoveries that were made. Okay. So let's look for a minute at just one experiment that Ethel Brown describes in her paper. So first of all, what is Hydra? Well, here's a picture of Hydra. It's a small animal. You can see it's about 10 millimeters in length. Lives in fresh water. It's radially symmetric. You can see the tube. And it has a mouth at one end with about 1 to 12 motile tentacles. So this slide now shows one of the experiments that she describes in her paper. And basically what she did is to transplant, to cut a piece off, cut one of those tentacles off from the mouth region and insert it to a different location and simply ask what happens. And what she observed is that she observed these outgrowths. So the key insight, or the most important thing that they did, was to utilize the fact that she had two different types of Hydra growing in the lab, one that was green, one that was white. And so what she did was to, in this transplantation experiment, for instance, to transplant a white tentacle and embed that into a green Hydra. And then she could ask, in that growth that results from around that transplant, what part of that growth derives from the host, that is the green animal, what part of it derives from the white, the donor. So this now is an actual photograph that she made, or a drawing that's in her paper, showing this spectacular and very important result for developmental biology. What it tells us is that this new tissue that grows out as a consequence of this transplantation includes both donor tissue, that's the white, as well as the recipient, the host tissue, which is green. Now that white tissue clearly remembers where it came from because it continues to make a tentacle. But that green tissue would never have made a tentacle under normal conditions. It has been induced to make these new structures in response to something, some effect that this white tissue has had on it. So this now I show in a pictorial form where you can see that the transplanted tissue, the transplanted tentacle, induces an outgrowth, and this outgrowth includes now both white and green tissue. This is an extremely important and fundamental observation. It was expanded upon and developed further by a German group run by a man by the name of Hans Spemann, who was studying amphibian development. And here we just show in pictorial a picture of a amphibian embryo that develops into a tadpole. And what they were doing, again, as Brown was doing, was to look at the behavior and influence of transplanted tissue. So they developed this idea further. And what they learned was that if they transplanted tissue, in this case from what's called the dorsal lip of the blastopore, this is an early embryonic stage, from the dorsal to the ventral side of the embryo, that they could explore issues of memory and influence. That is, did the tissue remember where it came from and made the same structure in a new ectopic location? And did it have any influence on the surrounding cells? And they found that, and this is an operational definition, they found that in some cases they could see that the cells would remember where they came from, but did not have any influence on the surrounding tissue. But there was one particular region, that is this dorsal lip of the blastopore, where if they transplanted it from the dorsal to the ventral side, they got a spectacular effect. And that's shown in this slide. That is, they could induce a new neural axis. So from this, they could conclude that these transplanted cells that induced a second neural axis must contain the property to induce these responses. That is, there are specialized groups of cells that produce inducers. And this is, again, a fundamental observation in developmental biology. And this was published in 1924 by Mangold and Spemann. So this tells us that there are paracrine inducers that exist and that influence what neighboring cells do, how they develop. That is, that there are groups of cells that produce inducers. We call these developmental organizers. We call them signaling centers. But then it became, if you will, the holy grail in developmental biology. One, to say, who are these inducers? What are they? How do they work? And how do they move across tissues? And I've illustrated that here in this little cartoon below. If we consider a hypothetical epithelial sheet, and indicating here just one cell, say that's making an inducer, indicated here in red. Somehow that inducer presumably spreads across the neighboring tissue to induce a response. That's the essence of paracrine signaling. The effort to identify what these inducers are consumed decades of work by many developmental biologists, but they were frustrated in these efforts, and for many, many decades, these inducers, these signals, their identity could not be established. But over many, many years, many experiments were done to really legitimize this idea that there are inducers that somehow must disperse across tissue to generate some sort of concentration gradient, and that the idea emerged that the position of each cell in that tissue is defined by the amount of inducer that it is exposed to. Lewis Wolpert, a British developmental biologist, developed this into a beautiful theory, and he coined the term positional information to describe this idea that the position of each cell is defined by the amount of inducer. But he also noted that if this distribution of these inducers is continuous, yet cells must be able to interpret that continuous distribution in terms of discrete responses. And he used this wonderful analogy of the French flag. Somehow this continuous distribution of inducer must be interpreted in fields of cells to generate discrete responses, in this case using this French flag as a way of illustrating that idea. This idea was further explored in this and described in this paper from Francis Crick. Francis Crick, of course, one of the two who discovered the structure of DNA. What Crick did was simply to ask the question, we know how large embryos are. We know the time it takes in development for them to do what they do, to develop. If these inducers simply move by passive diffusion from these signaling centers, from these developmental organizers, is there enough time for them to disperse across these fields if they move by simple passive diffusion? And he made some elegant calculations. The assumption that he made, because at this time we didn't know what those inducers are, he simply said, okay, I'm going to assume that these inducers are small molecules, maybe on the size of ATP, about 500 molecular weight. I'm going to assume, he said, that these small molecules are made and that they can move freely over and in and between cells. That is, they're not, cell membranes are not going to interrupt or affect their movement, that they are freely diffusible in and out of cells. And then he made these calculations based upon the presumed diffusion coefficient and other parameters. And the conclusion was that yes, there is enough time to generate these distributions of inducers. If there are these small molecules, they can freely diffuse in and out of cells. One of the resources that we have today, when I went to look up this paper, I didn't have to go to the library. I just went to an electronic database. And I asked the database, find a paper that had Crick and gradients in it. And it found this paper for me, and this is a figure from that paper that I've reproduced. But it also found a second entry in the database. And this apparently was a letter written to Nature in response to this article. I would never have been able to find this simply by going to the library. I don't even know if Crick knew about this letter, but I suspect that he did. I'm going to show you this letter here, and I'm going to read from it. It's entitled, Diffusion in Embryogenesis, Sir. This is from Elizabeth Duchar, who was a developmental biologist working in London at the time. As an embryologist who started work during the heyday of fields and gradients, I suppose I ought to be grateful to Dr. Francis Crick for allowing me a nostalgic look back at these long discredited concepts, which he has now resurrected, or should I say canonized, with the double halo of his own reputation and some elegant mathematics. There is, however, one point that he appears to overlook: the extreme rarity with which shear diffusion processes occur in living systems. 20 years ago, my better informed colleagues told me about active transport and permeases, and ever since then, if materials have diffused in and out of my experimental embryos, I've regarded it as a sign that they're dying or dead. A sheet of frozen dried tissue extended between source and sink might fit Dr. Crick's formula, but alas, it would not differentiate. Yours faithfully. I can only imagine what Crick's reaction might have been had he seen this. But recall that at that time, nobody knew what these inducers are. Today we do. We know that these inducers are proteins. And I show here a list of the proteins that we know constitute this language of development, if you will, that are these inducers that are responsible for these phenomena such as those I illustrated with the Hydra and with these amphibian embryos. There are proteins such as TGF-beta, or the Drosophila homolog decapentaplegic, or DPP, fibroblast growth factor, FGF, Hedgehog, Wnt or wingless, the Notch delta signaling system, the ligands delta serrate or jagged, and epidermal growth factor, EGF. So they're not small molecules like ATP. They do not diffuse freely in and out of cells. Unfortunately, Dr. Crick is no longer with us, but I have to assume that had he lived to see the day where these inducers had been identified and understood to be these proteins, he would have agreed with Elizabeth Duchar. So here's the point I want to make. These inducer signaling proteins constitute, if you will, the language of development. And that vocabulary, that small list of inducer proteins, is both small and it's universal. We can characterize, or we can show, that those same inducer proteins are important for the development of all various different tissues in insects, for instance in Drosophila, in mammals, and amphibians. It's the same list in virtually every organ and tissue, in virtually every animal that's been studied. And finally, of course, inducer signaling proteins cannot diffuse freely in and out of cells. So then the question arises, okay, now we know who they are, how do they work, and how do inducer signaling proteins move from producing cells to target cells? I've schematized that here in this cartoon. Again, red to indicate the inducer proteins. Somehow they must disperse across a field of cells to generate these gradients. I'm going to turn now to my favorite experimental organism, Drosophila melanogaster, and showing you this beautiful picture that was taken some years ago by E.B. Lewis, one of the great developmental geneticists who worked with Drosophila for many years, and he took this beautiful picture. We're going to focus on one region of the fly, that is the thorax, the dorsal thorax here, and the wings that are connected to that dorsal thorax. Both that dorsal thorax and some of the internal tissues, as well as the wing, are products made by the wing imaginal disc that grows during larval development, and we show that here in a mature larva. Now that wing imaginal disc has signaling centers, developmental organizers, if you will, and I show two of them in this slide, in this beautiful picture, where we see DPP, that TGF-beta homolog, is expressed in a stripe of cells that runs from the bottom to the top of the disc, as you can see here, and then wingless, also expressed in a beautiful stripe, and these stripes are orthogonal in this region of the wing disc. And many experiments done in many labs over the years have shown that both wingless and DPP function as inducer signaling proteins, and that these regions function as signaling centers in the wing disc. In fact, we can show, and this is a paper that and work that came from the lab of Marcos Gonzalez Gaitan, where they expressed a form of DPP in those DPP-expressing cells that was fused to the fluorescent protein GFP. And they could show that although the DPP was expressed in that line of cells, shown here in green, if they actually monitored and followed the distribution of the protein, that protein spreads out across the disk from the cells that express it in that line, and it forms this beautiful monotonic concentration gradient across the expanse of the disk. So the protein is expressed in the signaling center, but the protein disperses over long distance, and experiments done in many labs have shown that that distribution of DPP protein across the disk is essential for both the growth and the development of the cells that lie out at distances from that signaling center. So now we want to ask the question, how does that DPP move from the cells that make it to disperse and distribute across, for instance, in this case, the wing imaginal disc? So now I want to go step back a minute and review what we understand to be the three general models of cell-cell signaling. These are called endocrine signaling, neuronal signaling, and of course paracrine signaling, which is the topic of discussion in this presentation. Endocrine signaling, what is that? Well, we know that there are cells, for instance, in the endocrine pancreas that make insulin. Insulin is a hormone. It's secreted. It's released by those beta cells in the pancreas, enters the bloodstream, distributes throughout the body, through the bloodstream, exits the bloodstream to find target cells, receptors on those target cells to elicit a response. That dissemination of insulin is extremely rapid, and we assume uniform throughout the body. What I show here on the picture on the right, we also know that if for some people, patients with diabetes that cannot make insulin, that insulin can be supplied exogenously, for instance, with a hypodermic needle. And that insulin, once it enters the bloodstream, can also disseminate throughout the body. And the cells that receive it don't know, and they don't care where that insulin came from, whether it came from the pancreas or that hypodermic needle. That insulin functions in exactly the same way. So again, the characteristics of endocrine signaling are that it's rapid and it's uniform. Neuronal signaling. Well, we all know that neurons are these spectacular cells that make these asymmetric processes that can send and receive signals over long distance. And in this figure here, we're showing that at the end of an axon that's extended by a neuron, neurotransmitters localize and concentrate, and that they can be released to bind to receptors in the postsynaptic cell to elicit a response from this postsynaptic target cell. Now, the beauty of this system is its specificity. That is spatially specific, so a neuron does not simply release a neurotransmitter into space, but it releases at a pre-specified target. It's temporally precise, so it's released in a tightly temporally controlled manner. The signaling is extremely rapid, fractions of a second from the pre to the postsynaptic cell, and it's beautifully quantitative. And I simply show here a picture of a giraffe. If you think about those neurons that extend from the brain down into the body, or from the body into the brain in a giraffe, those are meters long. So the distance between the two cells, between the neuron and its target cell, can be meters. But yet the signaling can be spatially specific, temporally precise, rapid, and quantitative, because the signaling, the exchange of the signal, the release of that neurotransmitter is only at that pre-selected synapse, which is just nanometers, literally, away from its target. And let's contrast that now with paracrine signaling. How do we think about paracrine signaling? Paracrine signaling we think of as these secreted signaling signals, these secreted signals that are released by a signaling cell simply secreted into space. They move away from the signaling cell, from the producing cell, to find a receptor on some target cell. So the idea here, it's spatially limited because it's going to dissipate in space. It's temporally rather imprecise because it has to move by some random process, if you will, to find its target. It's relatively slow, and it's quantitatively imprecise. So in the context of development, where development needs to create these intricately and precise patterns and structures, the question is whether paracrine signaling can do the job, given that it's spatially limited, temporally imprecise, slow, and quantitatively imprecise. I want to just show one type of experiment that neurobiologists have done as they've used various experimental procedures to inquire and to investigate how neurons work. One of the types of experiments that they've done is to literally take a slice of a brain, then to make electrical recordings from that slice in response to neurotransmitter that's supplied by a pipette. And these experiments have been done many times in many labs by many investigators. And here's one type of trace that I took from a paper from just that type of experiment. So what can we learn from that kind of experiment? We can learn how neurons work, how they respond to neurotransmitter. These neurons have receptors for these neurotransmitters, and if that neurotransmitter finds that receptor, the neurons respond. Now, when neurobiologists do that experiment, they therefore conclude that the normal mechanism, the normal mode by which the neurotransmitter finds its receptor is simply diffusing freely in space? No. We know that the neurotransmitter is released only at a synapse and at that presynaptic terminal where those neurotransmitters are stored. That's the normal. Can we learn something by doing this type of experiment? Yes, of course we can. But that does not tell us the normal mode by which neurotransmitter arrives at those receptors in the postsynaptic cell. No one would ever suggest that. But now let's look at a very similar type of experiment that's done by developmental biologists. In this case, it's been shown that mouse embryos express an FGF, in this case FGF8. That's been shown to induce the expression of a target gene, if you will, called PAX9. And in this experiment, mouse embryos were shown to respond to ectopic FGF that diffuses from an implanted bead. And I show two examples of the experiment on the left, the control, and the right, the experimental. So what was done here was to take these beads, soak them in a solution containing a high concentration of FGF8, and then to take those beads and then plant them into a young embryo. Now, if those beads were simply incubated in PBS that had no FGF8, we can see the bead, as shown in the panel on the left, but no response of PAX9. If, however, those beads were pre-soaked in a solution that contained FGF8, then in situ hybridization to detect PAX9 messenger RNA showed that PAX9 is expressed in the tissue surrounding this bead. So what does this experiment tell us? This tells us that those cells around that bead can respond to ectopic FGF. They must have the FGF receptor. And if that receptor finds its ligand or is bound to ligand, those cells respond appropriately. But does that experiment tell us how FGF8 normally is released by cells that express it and moves to find cells that have the receptor? And I would argue no. So here now I show just a cartoon illustrating one model for paracrine signaling in an embryo. In this case, a cartoon for an epithelial sheet. If the cell in the middle is the cell that's producing the signal, these little red dots, we know, as I've shown you previously, that these signaling proteins can disseminate, can disperse across the field. One mechanism that's been considered is that, well, these standard classic paracrine signaling, they're simply secreted into space, and passive diffusion distributes them across the target field. So does this FGF8 soaked bead, is it consistent with that? Yes, it is. It's consistent with it. But does that experiment show us, does it tell us how the protein is normally, how that protein normally moves from the producing to the target cell? And I would say it doesn't do that anymore for this bead experiment that the developmental biologist does than it does for the neurobiologist who does the same type of experiment with neurotransmitter in that brain slice. So the question here is, does the diffusion of FGF8 from an implanted bead indicate that FGF8 normally disperses by diffusion? The answer is no. So diffusion may appear to be the simplest mechanism for dispersion of signaling proteins. But what I'm going to show you and describe in detail in the second presentation is that new experimental data now reveals that the mechanism by which these proteins move from producing to target cell is by a directed transport mechanism that involves exchanges at sites of direct contact. So in this slide, I'm showing these two what I call contrasting models for the dispersion of secreted extracellular signaling proteins. On the top, what we call passive extracellular diffusion. And as I've already described, this would be represented by the cell in the middle, which is making the signaling protein indicated by these red dots. And here the idea is that the protein is secreted into space. It diffuses, it moves by passive diffusion to disperse across the field, and then is taken up by cells in the field to generate a concentration gradient. That's one model. I'm going to contrast that with the model indicated in the lower drawing, which I'm going to call directed transport. And here, again, the cell in the middle is making that signaling protein. That signaling protein is dispersing across the field, generating the same concentration gradient. But the idea here is the protein is never free in the extracellular fluid. It moves along what are indicated these black lines here, which are actually specialized filipodia, which we call cytonemes. And the exchange between the producing and the target cell is at points of direct contact, much like a neurotransmitter, where the exchange is at a synapse, the regulated contact between the pre and the postsynaptic cell. What I'm going to describe in the second presentation is the data that underlies this model and leads to the following conclusions. So I'm going to call this the cytoneme directed transport and directed transfer model. Again, cytonemes are specialized signaling filipodia. We can show, and I'll show you the data in the next presentation, that cytonemes make direct contact with cells even at long distance. This can extend over dozens of cells. And we can show that cytonemes facilitate the exchange of signaling proteins at these sites of contact, and they transport signaling proteins between the cell bodies of the producing and the target cell.